metabolism. Simple pathways. Biochemical reaction. All living cells depend on biochemical reaction to maintain homeostasis. All of the biochemical reaction in an organism are collectively referred to as metabolism, which is of two basic types. Catabolic reaction that breaks down molecules, generally energy releasing or exergonic. Anabolic reaction that build new molecules, generally energy requiring or endergonic. Exergonic reaction provide energy for endergonic ones. Simple molecules such as glucose, amino acids, glycerol, and fatty acid. This is the example of ATP, ADP plus P. Heat release, catabolic reaction transfer energy from complex molecules to ATP. Exergonics, complex molecules such as starch, proteins, and lipids. Endergonic, anabolic reaction transfer energy from ATP to complex molecules. And heat release. All organisms, prokaryotic or eukaryotic, need to build the molecules they need and find the energy to do so. Metabolic pathways. Initial reactant. Pathway 1A, enzyme. Intermediates, C, enzyme 3, and final products is enzyme 4. They have 3, enzyme 1 to 3. This one the example of metabolic pathways. And pathway 2, they have enzyme 5, and the last F, enzyme 6 to G. Most biochemical reactions are part of a series of reactions referred to as a metabolic pathway. It takes multiple reactions to make end product. Pathway can be catabolic or anabolic. Each reaction is catalyzed by its own enzyme. Enzyme basic. Almost all biochemical reactions are catalyzed by a specific enzyme. Proteins that accelerate the rate of a reaction without being changed themselves lower the activation energy E. The need for enzyme provides a way to control or regulate biochemical reaction. Reaction 1 occur unless the enzyme that catalyzes the reaction is present and active. Enzyme lower the activation energy. Reaction 1 occur unless the E requirement is met. This one the example. Enzyme physically bind substrates. Control of enzyme activity. Biochemical reaction can be controlled by changes in enzyme activity, which can be influenced in several ways. Number 1, changes in the amount of enzyme or substrate. More enzyme and or more substrate, more product. Number 2, changes in temperature, pH or salt can affect enzyme structure, hence its activity. Number 3, availability of any necessary cofactors. Some enzymes don't work without a non-protein cofactor. Number 4, the last, effect of inhibitors, molecules that bind to enzyme and reduce their activity. Factors affecting enzyme activity. Temperature. Reaction occur more rapidly as temperature rises. As long as enzyme is active, heat can denature enzyme. And pH, enzyme structure depends on pH, affects charge or of R groups protein structure. And the third, substrate. reaction occur more rapidly as substrate rises. Saturation occurs when substrate is high enough. Enzyme denaturation. Enzymes are polypeptides that retain their ability to function only when folded properly 12 to 32. In temperature, pH, or salt can disrupt amino acid R group interaction, causing the protein to unfold, become denatured. Mutation can also lead to misfolded non-functional enzyme. Some enzymes require cofactors. Example, coenzyme protein proportion inactive plus coenzyme cofactor non-protein portion activator, and substrate holoenzyme whole enzyme active can be a metal ion vitamin or other non-protein. If the cofactor is organic, it is called coenzyme. Enzyme inhibition. Normal binding of substrate A substrate active site enzyme action of enzyme inhibitors competitive altered active site non-competitive inhibitor A, B, C, and C allosteric site. Inhibitors bind enzyme in one or two ways. Competitive inhibition binding to active site, allosteric inhibition binding elsewhere, changing shape. Inhibitors can bind reversely, can come off or irreversibly don't come off. Poisons. Feedback inhibition. The end of products of metabolic pathways are important, reversible. Enzyme inhibitors inhibit first enzyme in pathway turning the pathway off. Low inhibitor pathway on. High inhibitor equal pathway off. Can be competitive or allosteric inhibition. Provide an important way of regulating end product levels. ATP, adenosine triphosphate, preferred source of usable energy for all cells. Breaking bond of third phosphate releases ideal AMT of energy. Bond is easily broken, low E. This is why organisms convert food energy to ATP energy. How is ATP produced? In most organisms, energy from a food source is converted to energy in ATP by glycolysis followed by one of two processes of respiration, high ATP level. Glycolysis. Glycolysis is a catabolic pathway by which sugars such as glucose and several other food sources are broken down to 3 carbon molecules of pyruvic acid or pyruvate. Releases energy to yield to ATP for glucose. Also transfer high energy electrons plus each to NAD plus to yield to NADH. Oxidation reduction. Much of the energy in food molecules such as glucose is captured as highly energy electrons E negative by electron carriers such as NADH and FADH. When a molecule receives or gains electrons, it is said to be reduced. They are typically transferred as part of hydrogen atom. A molecule that gives up electrons loses H is said to be oxidized. Fermentation. ATP production begins and ends with glycolysis in organisms that ferment. Fermentation is all about recycling NAD+ so that glycolysis can continue. NADH is oxidized to NAD+ by reducing pyruvate to lactic acid. This one example. And respiration. After glycolysis energy in use in pyruvate and NADH is used to produce much more ATP by respiration. Krebs cycle breaks down pyruvate to 3CO2 energy captured as E by NADH and FADH2. Electron transport. From NADH, FADH2 used to produce H+ gradient. Chemosmosis, H+ gradient used to make ATP. The Krebs cycle, a cyclical metabolic pathway catalyzed by enzyme in the matrix of mitochondria, requires two C-acetyl groups connected to coenzyme acetyl-CoA. 3 negative C pyruvate plus C-CoA and 2 negative C acetyl-CoA plus CO2 Krebs cycle. Electron transport and chemosmosis occurs in the mitochondria of eukaryotes and at the plasma membrane of prokaryotes. Oxygen, O2, is usually the final electron acceptor, but other molecules can play this role. Anaerobic respiration. Lipid and protein catabolism. Lipids and proteins can also be used as sources of energy to produce ATP. Different amino acid enter glycolysis or Krebs cycle at various stages. Fatty acids are broken down to acetyl groups and feed into the Krebs cycle. The summary of ATP production: obligate anaerobes, fermentation or anaerobic respiration. Obligate aerobic respiration, and brief periods of fermentation. Facultative anaerobes can survive with IA via aerobic respiration or fermentation. All organisms depend on autotrophs. Autotrophs can produce molecules from CO2 and inorganic carbon source. All heterotrophs require an organic source of carbon. Organic molecules directly or indirectly come from autotrophs. The source of energy for autotrophic processes can be light. Photoautotrophs that carry out photosynthesis. Chemoautotrophs that use various molecules as a source of high energy light reaction of photosynthesis. Electrons from H2O energized by sunlight fuel the synthesis of ATP through electron transport and chemosmosis, much like respiration. Ultimately, reduce NADPH plus to NADPH. ATP in NADPH provide energy to fuel production of sugar in the dark reaction. Dark reaction involves an anabolic pathway known as the Calvin-Benson cycle. Endergonic reaction of this pathway are fueled by ATP and ADPH from the light reaction. The resulting sugars can be used as a source of energy or to build other organic